Hi, I'm Minoree Kohwi of the Department of Neuroscience here at the Zuckerman Institute, and it is my greatest pleasure to introduce to you Chris Walsh, our next speaker. He is the Chief of Genetics and Genomics at Boston Children's Hospital and a professor at Harvard Medical School and Howard Hughes Investigator. Chris is both a neurologist and a neuroscientist who is deeply fascinated by the genetics underlying cortical development. And he's an inspiration to many scientists in that he takes a dual action approach to his research. So on the one hand, he has a very hardcore basic biology approach, like a seminal work that he started as a postdoc with Connie Sepko, where he was one of the first people to develop a retroviral library to barcode neurons and then show that neuronal clones can scatter considerably across the cortex through development. And then on the other hand, a more patient-centric approach to map single gene mutations that underlie disease. And the output of this is that now he has identified dozens of genes that are mutated in various neurodevelopmental disorders. And some of these genes function in neurogenesis, and surprisingly, some of these genes are also targets of human brain evolution, which is really exciting. And most recently, he has succeeded in whole genome sequencing of single neurons from the human brain and showed that, and found that somatic mutations in subsets of these neurons can contribute to neurodevelopmental disorders and aging. And so with that, I'd like to welcome him. And the title of his talk is One Brain, Many Genomes, Somatic Mutations and Genome Diversity in Human Brain.
Well, thank you very much, Minoree, for that very generous introduction. And it's a real honor to be here to celebrate the opening of this new center, especially for a schlub like me from Boston who doesn't have any real clear connection to Columbia on his resume, but actually the connections are there. First of all, I just want to acknowledge the leader here who's been a mentor to me over my career, and that's Carol Mason. So she was a postdoc in Ray Guillory's lab when I was a student there, and she's always been my scientific big sister ever since then, and it's been a tremendous boost to me. But there are other connections to Columbia also. My very first summer job actually was at the Neurological Institute in the Department of Neuroradiology, and that, more than anything else, set me on a course to do neuroscience. And then my wife grew up in the neighborhood. I was married at the Riverside Church. And in fact, I'll tell you a little story about this very street corner out here, the corner of that left turn lane where you turn from Broadway onto 125th Street. On the day that I came here to ask my wife's parents for their permission to marry her, which was a thing we did in those days rather than just posting it on Facebook or Instagram, we decided to take a little drive out to Westchester. And so her father and she were in the first car, and I was in the second car with her mother. And we were driving down Broadway to make that left turn onto 125th Street. And just as we were coming up to the light, it turned yellow. And like anyone would, her father sped up to make the left turn. And I sped up right behind him to make the left turn. And then he suddenly realized that I was following him, and he stopped dead. And as in that moment, as I drove one of his Volvos into the other one of his Volvos, I looked out on the future site of the Forum building through a cloud of shards of exploding headlights and taillights. And I could have had no conception that many years later, here I would be talking to you all today. But anyway, today I'd like to talk to you about what's the difference between the genome of one neuron to the genome of the neuron next to it. And you would think that they would all be identical, and it turns out they're similar, both derived from the same zygote, but they have lots and lots of differences. And I'll be focusing really on single nucleotide mutations. And so this is the part of the talk where where the speaker tells you how wonderful their technology is. And I think most of you are probably familiar with single-cell RNA-seq, which is all the vogue. And single-cell DNA sequencing has a few cardinal differences. So RNA sequencing is increasingly rapid. You can buy a 10x machine. Single-cell DNA sequencing, where you take single neurons and sort the nuclei, and then amplify the entire genome and sequence the whole genome the same way you would do the genome of a blood cell, is extremely slow. This is increasingly standardized. This is technically very demanding. And this technology, by the way, was developed by two former students, Gilad Avroni, now an assistant professor at NYU, and Shu-Yu Kai, several years ago. This is increasingly high throughput. You can do, with DropSeq, you can do 100,000 cells in an afternoon. This is very low throughput. And this is more or less affordable. This is extremely expensive because every single cell you want to sequence, you have to do an entire genome. It's more than $1,000. So the question then is, why would anyone want to do this? But in fact, I think of it, the genome of these single cells, as really telling us, as this painting by Paul Gauguin suggests, where do we come from, what are we, and where are we going? And I'll tell you in brief in the next 15 or 20 minutes that the somatic mutations form a permanent forensic record of the entire pattern of cell divisions that generates our brain, that the clonal mutations can sometimes be important causes of neurological diseases, and that they probably impact the development of most of us. And then finally, that they can accumulate even after neurons have stopped dividing throughout life. So here's an example. These are a couple of neuronal genomes from a patient who died at 4 months old with no neurological diagnosis, and another person, a little baby who died at 6 months old. And we took single neurons, amplified their genomes, and sequenced it. And the surprising thing is we found that a typical neuron has between 300 and 900 mutations relative to the zygote of that same individual. How is that possible? And so to find that out, Rachel and Yan-Mei Do in Peter Park's lab basically took brains of about 75 people, 60 with autism and 15 controls, and did high coverage whole genome sequencing. And this allows us to capture the mutations, particularly in a systematic way for the first several cell divisions at least, and found that, in fact, an average brain has about 15 of these mosaic mutations just from probably the first 5 or 6 cell divisions alone. Detectable in more than about 3% of the cells of the body. The overall mutation rates are similar in normal brains and autism brains. The differences, which I'm not going to go into in the interest of time, are in the locations and the types of those mutations, which are more damaging in the case of the autism brains. But a typical brain, one of us, probably is very likely, if not certain, to have a clonal damaging exonic mutation in 2% or more of the cells in the brain, in some patch of our brain, which might be, for example, why people in my family tell me I'm so consistently challenged by social interactions. And this just shows and these are only the mutations we can detect. We have very poor sensitivity for detecting mutations fewer than about 3% of the cells, even with this extremely expensive high coverage sequencing. And what we find is that the first cell division actually is particularly error prone, has an average of 5 mutations in the first cell division of the egg, and after that, an average of about 3 to 4 mutations in every subsequent cell division. And so what that means is that when the zygote divides, every cell division carries with it 3 or 4 point mutations, plus occasional copy number variants, deletions or duplications, retrotransposon mobilization, and microsatellite mutations, so that by the time a neuron is born in the prefrontal cortex, because in most cases we study neurons of the dorsolateral prefrontal cortex, that neuron already has 300 to 900 unique point mutations, most of them functionally silent, but occasionally functionally damaging, and that the neuron next to it has a different 300 to 900 one, so that every neuron has a barcode of where it comes from. And then again, many neurons will have accumulated at least one clonal damaging SNV by the end of neurogenesis. So then the question is then, can we use these to understand how the human brain develops and compare it to what we know about the patterns of brain development from non-human species? And we know that the cortex contains 2 basic types of neurons, the excitatory neurons with long axons, the inhibitory neurons with short axons, and that these 2 neurons are extremely different in how they develop. The excitatory neurons are formed from the dorsal ventricular zone and migrate generally radially, as shown in this work here from Simon Hippenmeyer and Sunghai Shi, showing some clones of excitatory neurons. The non-pyramidal neurons, as described by Oscar Marin and Stuart Anderson and John Rubinstein, are not formed in the cortex at all. They're formed in the basal forebrain, and they migrate all over the place. And so they form these single clones, shown in blue or green, can be scattered, covering the entire cortex, as Minoree was referring to, that you can only see using these retroviral barcoding technologies. And so this suggests that maybe human somatic mutations can be used in a similar fashion. And so a very talented postdoctoral fellow, Peng Li, and August Yue, a bioinformatician, have been trying to make this work in humans by combining in a single cell the analysis of DNA with the RNA to tell us what type of neuron that is. And this shows some very preliminary results from their analysis. They've analyzed over 700 neurons from the prefrontal cortex, and we can actually see this divergence of excitatory and inhibitory neurons over the course of development. So that the different letters indicate different clades. These are large lineages of cells that diverge from early stages of development, and then there are various mutations within each clade that nest within each other because the higher number of mutations occur later during development. And so in this case, for example, and then this shows the identity of the cells. So the early mutations are present in a lot of cells, and in roughly the ratios you would expect, somewhat more than half of the cells containing these early mutations are excitatory neurons, and a little less than half are inhibitory neurons. And that's true for pretty much all of the different clades, that these early mutations label both types of neurons. But then for those clades where we have these later mutations that are present in progressively smaller numbers of cells, and where you can see that over here, in a figure published a couple of years ago, you see that, in fact, in this clade and this one, the mutations become increasingly limited to the excitatory cells because those mutations occurred after the two lineages diverged. In this clade, on the other hand, the mutations are becoming increasingly restricted to inhibitory neurons, although we don't have the very last mutations that would be completely restricted. And this, in fact, looks very much like an inhibitory clone. It spreads over the entire surface, almost, of the human cortex, but only in about 1 or 2% of the cells or less. Whereas this looks more like what we imagine an excitatory clone looks like, because it at least has some sort of coherence in the cortex. Uh-oh. And interestingly, we can determine roughly the threshold at which these clones become limited to excitatory clones. All of these mutations that are roughly limited to excitatory cells are only in about 2% of the cells, potentially suggesting that about 50 or so progenitors might generate an individual column of the cortex. We can also ask, okay, are the neurons in the cortex born in sequence, the way we know they are in mice and in monkeys? This is a figure from a paper by Pashko Rakesh from the 1970s, showing that in the monkey, the neurons in the bottom of the cortex can be labeled with tritiated thymidine injected at early stages of development, and then if you inject it later, the neurons being formed at those later ages occupy the more superficial layers of the cortex. And by using these nested clades of mutations, we can see that the early mutations label neurons that we know by their RNA transcripts are in all layers of the cortex, and then the later mutations are restricted to cells in the upper layers. Or here's another example from a different brain. Actually, we've analyzed now 3 different neurotypical brains for the cell lineage analysis. You see the same trend here. And this implies, in fact, that clones like this that extend over a couple of centimeters in the human cortex, in many cases, don't necessarily even go the full thickness of the cortex, suggesting that the clonal structure of the human brain allows considerable intermingling between clones derived from very distinct sources. Or you can see an example here where we have this whole nest of clones shown a different way. The late mutations are in these sort of 2 cells, and this late mutation is present only again, across about 1 centimeter of the cortex, but in fewer than 1% of the cells, and then earlier mutations occupy larger and larger cortical regions. So these somatic mutations then form a permanent forensic lineage map of the brain and probably the entire body. It has a lot of very wonderful features. It's immediate. It requires no transgenesis or any sort of intervention. It's permanent. There's no silencing of the marker, as we sometimes often see with retroviral markers. It's quantitative in that the DNA allele fraction actually can tell you exactly what proportion of different clones are generating various aspects of the brain. The mutations, in fact, are linked to DNA replication. So you can actually use the mutations to count cell divisions over the course of development. the mutations are dense enough, just the S and Vs alone, so that in principle, this map is complete. It's likely that virtually every cell division, or almost every cell division, is going to be marked. It's also archival, so it could be applicable to any human individual or any other species with a comparable mutation rate. So they say that Einstein's brain had this enlarged parietal cortex. Well, if someone brought me Einstein's brain and I could get some single cells out of that brain, we could potentially tell you why that brain was enlarged in the parietal cortex in terms of a lineage map. And then also this method is genomic. By its very nature, it correlates the pattern of development with the mutations in the genome that might actually be regulating or causing competition between clones. And so this then is harkening back to Gauguin's what are we in terms of being a clonal mosaic. And of course, sometimes these mosaic mutations can be damaging. In fact, not at all infrequently. So for example, these little abnormal clones in the brain, here or here or here or here, are called focal cortical dysplasias. And in fact, they are the most common cause of intractable epilepsy of children that results in surgery to remove them. And they are clonal marks, just like the ones I was showing you, except that instead of them being functionally silent, they represent gain-of-function mutations in the mTOR pathway by mutating a variety of genes. And in fact, but as I say, their shape and size is very similar to this functionally silent clone. But in fact, the person just had the bad luck that these happen to be occurring in the exome at those particular residues that cause functional changes in the clonal behavior. In fact, these focal cortical dysplasias, if you sort the cells into the neurons and the non-neuronal cells, the very smallest focal cortical dysplasias, like these two right here, the mutation is actually limited to the neurons and is absent in the glial cells, whereas the larger dysplasias like this that occupy an entire hemisphere, the glial cells shown here in light blue, as well as the neurons shown in purple, they both contain the mutation. So it suggests we can actually time approximately when and where this mutation occurred to a progenitor, which is relatively specified for neurons, which does not go on to form glial cells. And in fact, it's likely that these progenitors are the excitatory neuron progenitors based on mouse studies that I don't have time to go into. And in fact, mosaic mutations, based on our work and others, probably make up about 5% of cases of autism spectrum disorders. And we're hoping we can eventually map these mosaic autism mutations in the brain to learn what structures in the brain are involved by mosaic mutations that might be damaging enough to cause disease. Now, when we did that study on the newborns, we weren't just studying newborns. We've done the same experiment, taking neurons from the frontal cortex, from adolescents and adults and aged individuals, and even two genetically progeroid conditions associated with premature aging and premature neurological degeneration, and just took a handful of neurons from each case. We also studied the dentate gyrus of the hippocampus because this continues to generate neurons after birth, and so we thought it might have a different mutational pattern than the prefrontal cortex, where all of the neurons, I think by consensus, are postmitotic at the time of birth. And what we found, remarkably, is that even in older individuals, the number of mutations per genome continues to increase. So that in the prefrontal cortex, a typical neuron gains a new point mutation about every 2 weeks. And in the dentate gyrus, a typical neuron gains mutations about 1 every 10 days, so about 40 per year. And this is work of a former postdoc, Mike Lodato, and Rachel Roden again, and Mike Coulter. Now, you see that there's variability between individuals. Some people seem, this woman here, for example, seems to be living a very healthy life because her mutation counts are about the same as a teenager, other than the fact that she's dead, of course. And that even within an individual, there's some variability within individuals. Here's an outlier neuron that looks like it had some sort of oxidative catastrophes. It has about 7,000 neurons, many of which, mutations rather, many of which appear to be oxidative in nature. Remarkably, this rate of accumulation of mutations in neurons is almost exactly the same as has been recently reported for stem cells in the brain, in the colon, and the small intestine, and the liver. They report rates of about 35 to 40 per year. But in fact, now we know that most of these mutations accumulate not based on cell division, but just occur even in non-dividing cells. Now, we can understand more about these mutations by analyzing their mutational signature, which refers to the base pair substitution and then the 3 base pair context in which that occurs. And we know from the cancer world that ultraviolet light causes particular types of mutations, C to T changes in a particular context, or cigarette smoke tends to cause oxidative C to A mutations. And so this is seen in certain cancers and not in others. And obviously, ultraviolet light is seen in skin and head and neck cancers and not in lung cancer, for example. And so this same method of non-negative matrix factorization was applied by Daniel Kwan and Alison Barton in Peter Park's lab to our neuron mutations to see if our neuron mutations showed similar kinds of signatures or different sorts of signatures. And they defined 3 signatures, A, B, and C. Signature B, you can think of as birth mutations. They are present in those newborn neurons and don't accumulate appreciably in the prefrontal cortex. They do actually accumulate in the dentate gyrus a little bit. I'm not going to show you that data. They're almost always cytosine C to T mutations, probably due to cytosine deamination in the cell cycle. This is a familiar signature from cancer, one of the most common ones. And so these seem to be related to cell division indeed. Signature A, on the other hand, you can call an aging signature. This is absent in the newborns and goes up like a clock at about 20 mutations per year. It doesn't seem to care whether it's in the cells in the dentate gyrus or the prefrontal cortex. It even goes up at the same rate in these genetic causes of progeria. And so you might think of this as healthy aging, although it doesn't seem all that healthy. It resembles a cancer signature, cosmic 5, which is one of the most universal cancer signatures seen in essentially all cancers, and proportional to nothing other than the patient at the age of the cancer diagnosis. And again, generally assumed to be caused by some aspect of cell division, but now we know it has nothing to do with cell division because it accumulates in postmitotic neurons. Signature C, you can think, has a very broad nucleotide distribution, a lot of these oxidative C to A mutations. And so this resembles a cosmic signature 8, which has previously been interpreted as a failed DNA repair of oxidative damage to the genome. And so you might think of this as unhealthy aging. It's very variable from neuron to neuron. It's also absent in the newborns. And this makes us wonder whether this might be some remediable cause of mutation. Now, when we compare these signatures to the signatures found in the stem cells, it's amazing that even though this was generated by a completely different technology, the 3 principal signatures are almost indistinguishable. This age-related accumulation, which we call genocinium, it's some sort of aging of the genome, and no one really knows exactly what causes it. Or a proliferation-induced cause, which is congenital in the case of the CNS, or this oxidative cause, which again is shared between neurons and dividing cells in culture. Next, we looked at these progeric conditions associated with neuronal degeneration. These are 2 different genetic disorders, Cockayne syndrome and xeroderma pigmentosa, due to mutations in the DNA repair pathway. And in the case of Cockayne syndrome, these kids die when they're 5 or 10 years old, but they look like they're 80 years old because they have gray hair and they're gaunt and cachectic. And they have severe neurological degeneration. This particular form of xeroderma pigmentosa we chose because it also has neurological degeneration as well as a cancer predisposition. And so we thought, do the neurons in these kids look like neurons from an 80-year-old? And sure enough, they do. They have 2,000 to 4,000 mutations per genome, which is typically what we see in an 80-year-old neuron from the dorsolateral prefrontal cortex. We estimate that both of these conditions, the rate of accumulation of mutations is roughly tripled compared to the normal amount. And remarkably, we can define the particular signatures that are increased. In both of these disorders, it's the signature C, which is greatly increased, this failed repair of oxidative lesions. But also the disorders are a little different. This one also has an increase of signature B. But again, that signature A, that aging signature, is not increased in either of these genetic disorders, which is all the more remarkable when you think that these are disorders of DNA repair, that this aging signature doesn't even increase when DNA repair is faulty. So how does this all fit into the aging of our brains in general? And the way we think of it is something like this. So again, you're increasing. You have 2,500 mutations by the time you're 80 years old in the prefrontal cortex. You have about 4,000 mutations per genome in the dentate gyrus by the time you're 80 years old. And of course, these accumulate silently. Many of them are functionally are non-functional, but occasionally about 1 or 2% of them hit the exome, and about 1/3 of those are likely to be damaging. And so this process likely just goes on for years and years. And the lucky thing is, nature endowed us with 2 copies of most genes, and for most of the genes, we only need 1 good copy for it to remain functional. And so these mutations can accumulate silently with no obvious functional effect, and then eventually, along comes a mutation that happens to damage the second allele of a gene where the first allele was damaged 20 or 30 years ago. And in this case, you've created now a cell, just 1 cell, because these mutations are unique to that single cell, which is a knockout for an essential gene. And so you might think of this cell as being like a zombie cell, which I apologize, I know is also now the abbreviation for the Zuckerman Mind-Brain Behaviors Institute. And so these knockout cells accumulate, the mutations accumulate linearly, but because of the diploid genome, these knockout or zombie neurons actually accumulate exponentially. So there's a long period from which we're protected from them, but then they start to increase and increase more rapidly at later ages. And anything that changes the slope of that line, like these DNA repair mutations or whatever, has an exponential effect on the accumulation of these knockouts. Now, it's remarkable in terms of where we are going that we've known since Gompertz in the early 19th century that mortality is an exponential function. And so this shows mortality data from Australia from 2013, and you see that it's exponential. And in fact, all of the great diseases have these exponential shapes. And so what we're trying to figure out is whether this linear accumulation of mutations in a diploid genome might be a way of explaining this exponential shape of these curves. We know that, in fact, the accumulation of mutations in cells is what drives this exponential increase in cancer. And so we suspect that a similar sort of linear increase of mutations may drive exponential increases in some of these other diseases. So I'll just close by acknowledging the many people who've contributed to work that I discussed fairly briefly. Mike Lodato has really led the single-cell whole sequencing analysis. I'm having a little trouble finding his name in here, but here he is. And he's just starting his own lab at UMass Worcester. This was started by Gilad and Shuyu. Rachel and Sarah and Javier and Peng Peng Li has done the RNA and DNA. And at that point, I'll stop and take questions if there's time. Thank you very much.
Great talk. We have some hands up above, if you don't mind, first, and then 2 here.
Hi, thanks. Great talk. I have a question about, so allelic exclusion seems to be more frequent in the brain. How do you think allelic exclusion changes this effect of increasing mutation rates on the brain in particular?
I guess the effect would depend on how many genes would be involved by allelic exclusion and whether the genes which are subject to allelic exclusion would be functionally essential to that particular cell. For example, the olfactory receptors, their effect would depend on the particular cell in which they're expressed. And I don't know whether knocking out a gene and so that you no longer form functional protein from that locus, whether that would then have a downstream effect on regulating allelic exclusion itself. But the other thing about the brain is that there are a lot of genes in the brain which are dosage sensitive. A lot of the mutations associated with autism, for example, cause a disorder in the heterozygous state. So this simulation I was just describing, which simulates biallelic knockouts, actually could potentially underestimate their effect.
That's beautiful and a huge amount of work, so I feel bad asking this question, but I wonder if you've also looked in glial cells at mutations in glial cells.
So we have single cells sorted. It's a little expensive, even with used money. And so we haven't done single cell whole genome sequencing of glial cells. We have, however, done a little bit of sequencing of wondering, how common is it to be walking around with oncogenic mutations in your glial cells. And one of those 17-year-olds that I was showing, had a classic glioblastoma mutation in a little clone in the cortex. And so we already learned enough to know that there's a lot of stuff going on there, but we haven't really plumbed into it.
One more here.
Your C to T signature A showed an enormous amount of variability in the number of mutations at various ages. Can those be correlated with the cause of death or comorbidities?
It's quite possible that it may be. We do not have any actual data to be able to enlighten that. It does appear that different people accumulate. We don't have enough data to be able to prove that different people accumulate mutations because, at different rates, because we're only sampling a handful of cells, from a couple of different people. It appears that way, and it appears maybe that the dentate gyrus is more difficult or variable than the prefrontal cortex, but we don't have enough data to really be sure.
Does the widespread distribution that you see for your cortical cells argue against the Rikic radial unit hypothesis, or is it just that the mutations that would identify the clonal progenitors of those units occur too late for you to be able to detect them?
Oh, no, I think we have to completely rethink the radial unit. I mean, I think I was at a conference about this where I had an active discussion with Pashko on this very subject, but I think more fundamentally, I think electrophysiologists are reconsidering the whole columnar organization of the cortex, that the idea that there are columns that have boundaries is not something that Hubel and Wiesel intended when they coined the term, but instead electrophysiologists are starting to talking about laterally intercalated units. That the cortex is a continuous sheet that has a radial organization. The inputs come out and from the bottom and go out the bottom. And so it is radial in that sense. And so I'm not so sure, but I do think that it's not possible that humans develop according to some simple radial sheet. That we definitely have enough. We've looked at a lot of different brains at this point and a lot of different clones, and we don't see anything. We're analyzing now in detail the 17-18 border, which would be the best candidate to have some sort of clonal restriction. And there are likely to be some sort of clonal restriction, but tentatively we find that once a clone involves more than about 1% of the neurons, it spreads right across the 17-18 border. So that 1%, and that's like, that would be completely inconsistent with that.
Okay, one more there.
I was wondering if you have any evidence of the functional relevance of this accumulation. So maybe in animal models, looking at electrophysiological markers and trying to correlate with the number of mutations.
Yeah. We haven't done that. Of course, the question that jumps to mind about animal models is when a mouse dies at 3 years of age, does it die with 2,500 mutations in its prefrontal cortex like a human, or does it accumulate mutations at the same rate as a human? In which case, it might die with 200 or 300 mutations, and we have to find some other explanation for why those mice look gray and doddering, other than this accumulation of mutations. And I guess I just don't know the answer. We're trying to find out that answer. My suspicion would, in fact, be that the mouse looks like they're 80 years old, and so it probably has about 2,500 or 3,000 mutations, but that would then in turn imply that nature sets lifespan, and that signature A may play an important role in doing that.
Thank you so much, Chris. Appreciate it.